So as I said in a previous video, and I just kind of wanted to rehash this and clarify anything, and that is amino acid metabolism. What does it consist of, and where does it take place, and everything like that, and why is it taking place? And whenever I say metabolism, in this case, I'm talking specifically about catabolism, so the breakdown of amino acids. So amino acid catabolism is quite complicated. It's got some common features among different amino acids and how they're metabolized, but then there are also some unique aspects. For instance, the amino acid serine and threonine. Well, they are going to use an enzyme known as serine threonine dehydratase. And what that enzyme is going to do is it's going to produce free ammonium. Okay, now what's the fate of that free ammonium? We'll get back to that in just a second. The amino acids alanine and cysteine, well, those are two different amino acids that are going to use amino transferases. Now, as they use amino transferases, what they are both going to do is they're going to make glutamate, whereas serine and threonine are not going to make glutamate. Now, essentially what you can do is you can look at what's taking place in a couple of different tissues. What we really want to do is we want to get ammonia into urea. And we do that through the urea cycle. This takes place in the liver. Okay. Now, by taking place in the liver, this urea can ultimately, as we've said previously, make its way into the blood, then make its way to the kidneys, and then once in the kidneys, it can be excreted in the urine. Okay, and we have to do that because this ammonia can be toxic. Okay, now what we want to do is look at our different tissues. Let's just classify them as muscle tissue, liver, and others. Now, the connection between all of these is your blood. And so what we need to have is we need to have molecules that can actually be transported through the blood. Okay. Now, glutamate as a starting molecule in other tissue, well, what we can do to that is we can put some of our NH4 onto it and make glutamine. Glutamine can be transported through the blood into the liver. Now, once in the liver, well, it can be deaminated through the enzyme glutaminase. I should have planned for more space there. And glutaminase is going to make glutamate. And it's going to release NH3, or NH4 as it would be. That NH4 can be integrated into the urea and follow the previously described excretion pattern, or course. Now, the enzyme that kind of kicked this all off was glutamine synthetase. Okay, so now if we go over to muscle tissue, well, we can use an amino acid and use an amino transferase, as is the case with like alanine and cysteine. And what we can do is transfer our NH4 from here, or NH3 from here, onto alpha ketoglutarate and make glutamate, GLU. Okay, that glutamate can serve as the molecule where we do a transamination reaction, a PLP dependent transamination, a second PLP dependent transamination where we make alanine. Obviously what I set up here, we wouldn't necessarily want to transfer an amino group from an alanine ultimately to an alanine, but we can transfer our amino group from glutamate onto a pyruvate molecule in order to make alanine. That alanine can go through the blood to the liver, where we still have alanine. And this is known as the glucose alanine cycle. Okay, that alanine in the blood, in the liver, will then undergo another PLP dependent transamination reaction, and we're going to input alpha ketoglutarate to produce glutamate, and from this alanine we'll produce pyruvate. That pyruvate we can use for gluconeogenesis, and make glucose. Therefore we have our second molecule for our glucose alanine cycle. That glucose can go back into the muscle tissue, or it can be broken down into our alpha keto acid. In this case, our alpha keto acid is pyruvate, okay, through glycolysis. Okay, now this glutamate, what we're going to use for this is we're going to, in the liver again, that glutamate will use GLU dehydrogenase to generate our NH4 plus that we can then integrate into our urea, and therefore we've excreted it. So what varies a little bit here is if you're talking about muscle tissue, you're going to take an amino acid and you're going to do some transamination reactions. If you're talking about in any other cell type or any other tissue type, chances are you're going to use the enzyme glutamine synthetase, because that glutamine can enter the bloodstream and be transported to the liver, where you can use glutaminase, release that NH4. That NH4 can then go into the mitochondria using to make our carbamoyl phosphate, and then enter the urea cycle to generate urea and be lost, or released as it would be. If you're in the muscle, chances are what's going to happen is you're going to do a transamination reaction where you remove that nitrogen, or you simply transfer any free ammonium or ammonia onto an amino acid, or onto alpha ketoglutarate to make glutamate. So what I wanted to do with this was just kind of clarify that these processes do take place, and it's important to know what your sources are. So in the case of serine threonine dehydratase, well, that is going to produce free NH4. Well, can this be used in muscle tissue? Can you metabolize serine and threonine in the muscle? Yes, you can. That NH4 is going to be added onto a PLP molecule to be added to PMP, so that it can ultimately be added to alpha ketoglutarate to make that glutamate. All right. Well, I hope this is helpful. I think that it's important to kind of draw these pathways out and try to figure out, well, what is this, what are some of the important parts of it all? Some of the important parts of it all are that PLP and understanding a transamination reaction. All right, well, thank you very much. Have a good one.